Welcome, everyone. We're going to wait just 1 or 2 minutes here to allow everybody else to log on, and then we'll get started. I see the participants' numbers are increasing, so we'll just wait another 30 seconds until they kind of level off, and then we'll get started. All right, so welcome. This is our panel discussion today about colorectal cancer research at Vanderbilt. My name is David Cortez. I'm the Associate Director for Basic Sciences in the Vanderbilt-Ingram Cancer Center and the Interim Chair of Biochemistry in the School of Medicine. So about 1.9 million Americans are diagnosed with cancer every year, and over 600,000 will die of this disease. About 50,000 of those will die of colorectal cancer. And it's the fourth most common cancer diagnosed in men and women in the US. In fact, about 1 in 25 Americans will be diagnosed with colorectal cancer in their lifetimes. So the good news is over the last 2 decades, the rates of death of colorectal cancer have been falling. In fact, about 2% every year for the last 20 years. However, it's still a major challenge, obviously. There's actually been an increase in young people being diagnosed with colorectal cancer, and the 5-year survival rate for this disease is still only 65%. So today we will discuss what researchers at Vanderbilt are doing to improve these statistics. This event is hosted by the School of Medicine Basic Sciences. If you have any questions, please go ahead and put them in the Q&A, and we will get to them towards the end of this moderated discussion. And before we begin with our panelists, I'm going to turn this over to the organizer of this event, Erin Conley, who is a survivor of colorectal cancer, for a brief survivor story.
Thank you so much, Dr. Cortez. As you said, my name is Erin Conley. I work in the School of Medicine Basic Sciences as the Director of External Affairs and Partnerships. In 2013, when I was 27, I was diagnosed with stage 3 colon cancer, which was definitely a shock as I was a healthy young adult up until that point. Prior to my diagnosis, I started feeling fatigued. I started having chest pains and heart palpitations, blurred vision, and I knew something was going wrong. I went and got a physical. I learned that I had less than half the amount of blood I was supposed to have in my body at the time, and through some other tests was led to get a colonoscopy. In that colonoscopy, they found a tumor that was bleeding, and that's how I was losing blood, and I was diagnosed with cancer at that point. I then had a major surgery to remove 8 inches of my colon and a fist-sized tumor, as well as 32 lymph nodes. 5 of those were cancerous, which is what made it stage 3 and led me to getting chemotherapy. And I went through 6 months of chemotherapy through a regimen called FOLFOX, which is every 2 weeks for 49 hours. Definitely there's a good number of side effects from that around nausea, neuropathy, headaches, fatigue, etc. And in this time of my treatment, I was actually able to participate in a research study on the genetic predispositions for early onset colon cancer, such as Lynch syndrome. And through that experience, I got vastly interested in how research, especially basic and translational research, can impact new treatments and patient outcomes and patient experiences, which really led me into the work that I do now. I have been lucky enough to have great health ever since I finished chemo in June of 2014, and I'm currently treated at the Ingram Cancer Center. My doctor there is Dr. Siembor, who's wonderful, and feel very grateful to be here today and really excited to learn more about the research that's going on towards new personalized targeted therapies, immunotherapies, et cetera, that will be less toxic and have great outcomes for patients. So thank you, Dr. Cortez.
Thanks, Aaron. So let's introduce our panelists. Why don't we start with Bob? You're in my upper right corner, and we'll go around. Each of you just introduce yourself and tell us about what you do.
So my name is Bob Coffey, and I'm a physician scientist who has been at Vanderbilt for nearly 35 years now. And I'm direct the GI Special Program of Research Excellence, which is a very coveted award from NCI. These are organ-based programs that deal with the nuances and intricacies of cancer in various different organ sites, for example, breast and lung. And I've been very honored to be able to have led the Vanderbilt GI Sport, which has been continuously funded since 2002 and is focused entirely on colorectal cancer. So that is it in a nutshell.
Thanks, Bob. Alissa?
Sure. So I'm a professor of cell and developmental biology, and I've been at Vanderbilt since 2003. My laboratory studies basic biologies of how cancers become aggressive and spread throughout the body. And about 12, 13 years ago, we became interested in the field of extracellular vesicles and how they drive cancer aggressiveness. These are small vesicles that carry protein and RNA components. So a couple of years ago, we started a program at Vanderbilt in extracellular vesicle research that's going to be turning into a center in July 2021. My laboratory studies many kinds of cancers, but we started working on colorectal cancer to understand how extracellular vesicles can transfer not only proteins, but also RNAs between cells and influence the tumor microenvironment and tumor growth and aggressiveness. And so recently, together with Dr. Coffey and other investigators, we were funded with a large program project from National Cancer Institute to pursue this work on how RNAs are transferred between cells in colorectal cancer and how that drives aggressiveness of that cancer.
Thanks, Alissa. Jordan?
Hi, my name is Jordan Berlin. I'm the representative clinician. I'm a clinical oncologist and a gastrointestinal cancer specialist. I've worked with colon cancer through my entire career. I've also along with Bob, actually, Bob was once the co-director of the GI oncology program with me. And now Bob and I work together on the GI SPORE, where I assist him in running the administrative core and co-direct one of the projects on the SPORE. And I'm also the associate director for clinical research and run our experimental drug program here at Vanderbilt. So I've got my hands in a lot of pots working on things. In particular, colon cancer has been a big part of my life.
Thanks, Jordan. And Ethan?
Yes, my name's Ethan Lee. I'm a professor of cell and developmental biology, pharmacology, and cancer biology. And I'm a basic researcher. My lab studies this cell-cell communication pathway called the WIT pathway. And as it turns out that it's inappropriately activated in over 90% of all colorectal cancers. And so there's a lot of effort to try to understand this pathway. And the idea is that if you can target it, you can use these drugs and to treat colon cancer. And that's where I come from.
Thanks, everyone. So I'm going to start with just a simple question, and I'll pose it to Jordan. What is colorectal cancer, and how do people get it?
That's a great question. So colorectal cancer, the thing we were really talking about is something called adenocarcinoma of the colon. There are other rarer forms of colon cancer, but we're really talking about adenocarcinoma of the colon. It is cancer that grows out of the glands that make up the colon and the glandular cells, and the development is not entirely clear. We do think that in what we call sporadic cases that aren't related to genetics, there may be some effect of bile acids. There are a lot of other possible inciters, but of course the biggest cause is aging normally. The second largest cause of this cancer seems to be a genetic cause, and there are several genetic family syndromes. The most commonly known are something called the Lynch syndrome, or we call it HNPCC, as well as familial adenomatous polyposis. People will have a lot of family members. The familial polyposis, the patients will have a lot of polyps in their colon. And now there's an increasing interest in the serrated adenomas, which Bob will be able to talk a little bit more about. The serrated adenoma syndromes, personally, that's my family. We have it run in our family. We have 4 members of the family affected by that already. And that the genetic causes are an important one, especially if you have multiple family members involved. It's always worth talking to your doctor about seeing genetic family counselors. The nice thing about it is a disease that we can detect early by doing colonoscopy. But as Aaron demonstrates, we start colonoscopy now, recommended at age 45. But we're seeing this early onset colorectal cancer, which is becoming much more common. And we're not screening those people routinely because it's not something we would recommend. It's still rare among the younger people, but it's rising. And so that's a big problem. And for that, we actually brought in Kathy Ang, who, along with Kristen Siambor, Dr. Connolly's doctor, is that we actually have a specific clinic and work going on specifically looking at young people with colorectal cancer.
One thing that I might add is that Jordan alluded to this serrated adenoma new entity that's been recognized more recently. And we were fortunate to be able to get one of Joe Biden's Moonshot Initiative grants. And this was looking to create a single cell atlas of the 2 most common pre-malignant tumors that one sees in the colon. And the first is the very well-known adenoma, and that is driven by stem cells and by Wnt signaling that Ethan will be able to tell us more about. But we've just discovered that the other more common pre-malignant lesion is what is called a sessile serrated lesion that can give rise to these microsatellite unstable right-sided cancers. And we've just learned that by doing single-cell RNA-seq and then multiplex immunofluorescence and considering the microbiome, that these sessile serrated lesions don't derive from stem cells. Rather, they derive from differentiated cells, and they exhibit a gastric metaplasia. That is, that the differentiated cells in the colon now take on features of the stomach mucosa, and that is a definition of a metaplasia. And there's surrounding inflammation and activation of an oncogene called BRAF. And so we're delighted to say that that paper is under, we're resubmitting to Cell, and we think there's a very good chance that it will be accepted there. So that's something new on the research front, in which we're identifying a new etiology of the second most common cause of colon cancer.
So it sounds to me like that colon cancer is not one disease. It's actually multiple different diseases. Can one of you kind of expand on that? Maybe Ethan?
I think I'm gonna have to pass on that one. Maybe Jordan can.
I'd be happy to answer. So we're actually, It's interesting, when we develop as humans in the fetal stage, we actually have 3 hollow tubes that kind of form all our organs. The foregut, the midgut, and the hindgut. And actually the colon is formed partially from the midgut and partially from the hindgut. And that may play a role in how this is at least a couple different diseases, in that we are seeing that the right side of the colon, the cancers are very different than the left side of the colon. The prognosis, the response to therapies, and the changes in the genes of the cancer. They might not be the changes in the genes of you or the person, but the changes in the cancer can vary from the right side to the left side. Even within that, of course, if you have changes in certain genes, it may make you more susceptible to certain therapies in your cancer, that is. So we're seeing a lot of kind of subdividing of the disease. We now look at it as more than one disease. So what we're doing is making a common tumor into a bunch of rare tumors or less common tumors by looking at it as on an individual basis. But that's also our therapeutic treatment of these diseases. We're able to individualize therapies. Not everybody gets 6 months of therapy as Aaron did anymore. We were actually learning who can get 3 months of therapy instead of 6 months of therapy. So we are definitely trying to individualize, and as we do it, it's because the cancers are very different in the molecular standpoint, and again, in their origin, sessile serrated adenomas versus regular adenomas versus those that we don't necessarily associate with the polyp, although most of them come from one of the 2 types of polyps, adenomas or sessile serrated.
Okay.
I would also say the explosion of gene expression profiling has helped us identify subsets of colorectal cancer. And so they're now the CMS classification, which is broken into 4 different subtypes of colorectal cancer, each with distinguishing features based on bulk, taking the whole tumor RNA-seq. And that is now an increasingly accepted classification system for colorectal cancer.
Okay. And Alissa, you mentioned in your introduction something about microenvironment. And we heard about pre-malignant tumors and the tumor itself, the microenvironment. What do all these things mean? What is going on when somebody has a tumor that's developing?
Yeah, so, something that's really been realized over the past few decades is that our original concept of how cancers develop is very simplistic. So we used to think of just gene mutations happening in the cancer cell as driving everything. And clearly that's important. So, mutations, if you get the right combination in key signaling pathways that those tumor cells depend on, that will drive the tumor, but it's much more complicated from that because of the interface of those tumors and the pre-tumor cells, like the colon epithelial cells with the microenvironment. And in fact, the colon is a really interesting case because, because it's managing food and nutrients, and it's filled with bacteria. It's really an environment. The epithelial cells are interacting not only with normal other kinds of cells like the immune system, but also with bacteria, bacterial products. One of the risk factors for developing colon cancer is patients that have autoimmune diseases in the colon, like inflammatory bowel disease. And so there's this kind of complex interplay between what's driving proliferation and survival of the epithelial cells with all these other microenvironmental factors.
Thanks. So Aaron mentioned that he received FOLFAX therapy, I think is what he called it. Is that the standard therapy right now, and is the only available therapy, or how does that work? Is that what people are being usually getting? Jordan, maybe you want to take that as a clinician.
And again, I was getting a page. I apologize.
It's okay.
What were you asking again?
I was asking Aaron mentioned he received Fulfax therapy.
Yeah.
What is that? Is that what most patients get?
So yes, so it's interesting. We have a limited. We actually still mostly focus on chemotherapy in this disease, although we have what we call some of the more modern therapies. But a lot of it is chemotherapy, and in fact, actually, what we have are a few different drugs that the French have come up with some very interesting regimens. One of which is Fulfax, and what they did was, rather than use all the names of the drugs, they came up with a kind of scrunching of them together. They borrow a few letters from each of the drug names, and so Fulfax is actually folinic acid or leucovorin. Folinic acid is related to folic acid. 5-FU, which is just an F, in that, which is 5-fluorouracil, which is our oldest drug. Not a swear word, I swear. And oxaliplatin, which is a platinum-based agent with a very interesting molecular structure. And that combination is the most commonly used in what we call adjuvant therapy, or treatment of people who've had surgical resection of all visible disease, for whom we're worried that cancer might come back. To help prevent the cancer from coming back, we try to kill what's called the micrometastatic disease, or very small metastases too small for us to see, by adding the chemotherapy. Fulfax is the most common regimen we use. We also can use, instead of the 5-FU and the folinic acid, there's a pill form called capecitabine that gets converted to 5-FU in your liver and in your tumor cells. And we will sometimes use capecitabine and oxaliplatin as the regimen of choice in some people. But the majority of patients with stage 3 get one of those two regimens. Some stage 2 patients get chemotherapy, and stage 1 resected. That's a very early stage. They don't need any further therapy.
And why do these therapies work sometimes, but not for everyone?
That's a great question, and probably $1 million question. But the fact is that what we see is that cancer cells can develop resistance mechanisms to chemotherapy. And in fact, oxaliplatin, part of the reason we use it is because it overcomes some of those resistance mechanisms, so that when we give chemotherapy, it basically kills fast-growing cells. However, the 5-FU, which is the mainstay of the therapy, binds to something called thymidylate synthase. So one of the things that tumors do is they develop too much thymidylate synthase, too much of this protein, so that it overwhelms the amount of 5-FU we can give. We can't give enough to block all the thymidylate synthase, and the tumor cells get around it. Oxaliplatin helps reduce that, but it doesn't reduce it in 100% of the tumor cells. So if they have that resistance mechanism, they can grow and survive despite. But probably the biggest reason they survive the chemotherapy is something called tumor dormancy. Tumor cells stop growing, they become dormant, and that actually makes them resistant to chemo. Over time, the chemo gets thrown out, but the cells haven't been trying to divide. They haven't been using the DNA we're trying to harm. And somewhere along the line, something switches on and the cancer starts growing again. And then it can come back anywhere from a few months to up to 5 years out. We have seen few cases beyond 5 years, but most of the colon cancer cases, if they're going to come back, they're going to come back within the first 5 years. And in fact, we have data on 25,000 patients that 80% of those that will come back in the first 3 years.
I just might add to that, and it's commonly thought that the conventional chemotherapy, which is going to hit, say, the bulk of the rapidly proliferating cells, but the stem cells, which tend to be less proliferative, are the cells that may enter into what Jordan said was dormancy. And one of the projects in our SPORE, myself and Ken Lau in cell and developmental biology, are trying to determine stem cell populations in the mouse that may be repopulating the tumor. And so there are two very common stem cells in the colon. One is called LGR5 and the other is called LRIG1. And we've been able to make mice that have either green for LGR5 or red for LRIG1. I might say within our SPORE projects that are all basic science driven, but trying to achieve a translational outcome, we have a patient advocate as an integral member of each of the projects. And I should mention that our patient advocate on this particular project is Ron Obanoff, and he made it possible for us to generate this mouse that wherever LRIG1 was being made, we saw red. And so now we're able to create tumors in the mouse, treat them with these drugs like Folfox that Jordan mentioned, see the tumor shrink, and now we can see, okay, what cells are going to repopulate that tumor? Are they going to be the red cells or the green cells? And now when we determine that, and we think it is now the LRIG1 cells, that we can begin to characterize those and see if they have a vulnerability that we could then target that particular stem cell population that would then prevent the recurrence of the tumor. So that's a project one in our presently funded GI SPORE.
I'd like to add one other thing just to brag about Vanderbilt's involvement. Folfox includes oxaliplatin. The approval of oxaliplatin came from a registration trial that was written and led by Mace Rothenberg and I from Vanderbilt. So that drug got its FDA approval through Vanderbilt's leadership.
So this brings us to the point where we need to start discussing the basic research that's trying to look for new targets. It sounds like, Bob, you said that we have to figure out how to kill these stem cells that remain dormant and then start repopulating. And Ethan, you introduced yourself as saying that you were studying the Wnt pathway, which may be a target. Can you expand on that? How is the Wnt pathway? What is the Wnt pathway? Is this a target for these repopulating cells, or would it be a better drug target than what we're using now?
Yeah, so the Wnt is actually a contraction of two words. It's wingless, which is originally identified as a fly mutant. They're missing wings, and actually you can find them in pet stores because they're used to feed fish and reptiles. And the other word is int1, which was identified as a proto-oncogene. And in order to make so you have people studying wingless in trying to understand development in flies, and int1 trying to understand how cancers form. And to make it easier for the two areas in biology to communicate with one another and share information, they decided to make it Wnt. So Wnt is a contraction of wingless and int. And it turns out that the Wnt pathway, which is a cell-cell communication pathway, it's a predominant growth pathway in the intestine. So your intestine, the lining of your intestine slough off every 5 or 7 days, and it must be replenished. And it's replenished through the activity of these stem cells, which divide, and one cell becomes a stem cell, the other one becomes the other cells in the intestine. And so it turns out that in colon cancer, mutation in this pathway is very, very common. And so you can think of it as being stuck in the on position. So what happens is that these cells they continue to divide uncontrollably. And one of the protein that was identified very early on is called APC, which Jordan kind of mentioned, which it stands for Adenomatous Polyposis coli. And it's mutated in over 80% of all colorectal cancers. So if you have a polyp removed from your intestine, there's a very good chance that it's due to a mutation in APC. And it's often called the gateway to colon cancer. So the idea is that if we know how we can fix that, then presumably we can treat the vast majority of colorectal cancer, because there are other mutations in the pathway that leads to its inappropriate activation other than APC. And so it's been estimated over 90% of all cases of non-hereditary colorectal cancer has an activating mutation in this pathway. So the idea is if you inhibit it, you can possibly use it to treat colorectal cancer.
Would this be a treatment that we would substitute for the chemotherapy, or would it be used after the chemotherapy or what?
Well, it could be used in conjunction with the other therapy, but it could be used, I guess, for treatment. But I think Jordan and Bob would have a better sense of that. One of the things that's really that have come my own experience is that really understanding how it's like a car, right? You can't fix something unless you know how it works. And very early on, when I was studying this pathway, we developed actually a mathematical model of the pathway, which as far as therapy you can think of. It turns out that the people in Novartis noticed it and then used our model, then tried to develop drugs to target the Wnt pathway for treating colorectal cancer. And they came up with a class of drug called tangerase inhibitors. It was in preclinical studies at that time, and I don't know how far it's gone, but it was really first demonstration that you could use these kind of very basic models developed from basic science and to develop therapy to treat a disease. And I think it was the first example of that. We felt really good of, doing very basic science. You can eventually lead to some kind of treatment.
And you have a drug development effort in your own group as well, don't you?
Yeah, we we were studying the Wnt pathway, and we took advantage of Bob's GI sport very early on. He dangled money and a possibility of doing very exciting science in front of us, and we took it. And what we did was we decided that we could use our system that we were developing actually in frog eggs of all things to look for chemicals that could maybe target the pathway, that we could potentially use as tools to study the pathway, but also potentially be sort of the basis for maybe developing drugs against the pathway. And what we found was this compound called Provinium, and which is an old drug. It was used in the 1950s for treating pinworm, and we found that it was a very effective inhibitor of the Wnt pathway. And we, to make a long story short, we studied this drug, and we took it a little bit further. We were able to get the FDA to designate it as an orphan indication for FAP, familial adenomatous polyposis. So, which is a precancerous condition that Jordan mentioned earlier. And so that was one example of how just trying to understand basically the Wnt pathway, a curiosity-driven science, could lead to the development of therapy. And I would like to mention at this point that so most of us where I am is we do basic science because it's basically curiosity-driven science. We just want to know how things work and why things are the way they are. And however, a lot of times the answer to these questions have led to the most spectacular and far-reaching consequences in the treatment of disease. Think CRISPR, that many of you may already know, in the COVID era. If you get a test, it would not have been possible without the discovery of this protein that was discovered in a vent bacteria, that it can survive really hot temperatures, and that led to the development of PCR. It has revolutionized medicine. And even the Moderna drug and the Pfizer drug that many of you may have received, the RNA therapy that was developed, people were studying how RNAs can be stable outside the cell. And those are really basic science questions that were being studied decades ago. And it wasn't until obvious, that until now, that we could use that information then to develop treatment. So a lot of times basic science takes a long time, but the result can be quite spectacular and has far-reaching consequences.
So this is you mentioned this basic science leading to really important discoveries, and perhaps we should take a step back here and really define what is basic science. Alissa, would you consider yourself a basic scientist, and what does that mean?
Yeah, I am a basic scientist, and I think Ethan actually did a really good job at introducing this concept. So a basic scientist is someone who is driven by curiosity and wants to understand the fundamentals of how things work. So I would say, and so that means it's not necessarily driven by, I want to cure this disease, but often, as Ethan mentioned, later on, other people find a way to use that information because it is so fundamental in making new therapies. So for my own work, I would say that we actually like to go back and forth between normal systems and disease, because we believe that the disease informs normal biology and normal biology informs the disease. And I think that Ethan's example of trying to understand how signaling happens in, frog oocytes and Drosophila is a great example of how those systems are conserved in humans and deregulated in disease. And disease shows you where those key points are that can be broken and are interesting to study both from a disease standpoint and from the normal standpoint.
And your own research, I know, is on something called extracellular vesicles. So can you explain, you mentioned that in your introduction, can you just explain what is a vesicle and what does it mean and why is it relevant to colorectal cancer?
Sure. So for many years, we've known that cells communicate with each other through secreted factors. And we thought that those were all sort of soluble protein type factors. And what was realized a couple of decades ago, but actually largely ignored until about 10 years ago, was that actually there are these vesicles that are released from cells. They're sort of like mini cells in that they have arrayed on the outside of the vesicle many of the same components, proteins and lipids that cells have arrayed. And on the inside, they have things like cytoplasmic proteins and RNAs. And it turns out this is a way to transport and protect those components that otherwise couldn't be transmitted between cells. And RNA has been a particularly interesting one among all those cargoes that extracellular vesicles can transmit, because they have the potential to alter gene expression in recipient cells and really fundamentally change cell phenotypes.
And Bob, I know your research also involves extracellular vesicles, and as part of the SPORE, you're taking these basic science discoveries or knowledge, increasing that knowledge, but then also trying to use it more in the clinic. So can you expand on how, for example, the knowledge about extracellular vesicles might be translated eventually to help patients?
Sure. I could talk probably about an hour on that topic, but I'll give you just a couple very short vignettes. One is that we've actually found the EGF receptor on these extracellular vesicles, and we've developed reagents where we can tell on that vesicle the amount of EGF receptor there and its activity state. And so we can potentially use that as a biomarker because certain cancers overexpress the EGF receptor. And sometimes it's even in a mutant form in the case of brain tumors. So that's a potential, a biomarker. We've been delving deeper into what's produced by these extracellular vesicles. And one molecule that recently has really caught our attention is a molecule called CD73. So this is on the surface of colorectal cancer extracellular vesicles. And it turns out that this is a ectonucleotidase of all things. And what it does is it converts AMP to adenosine. And it turns out adenosine is an important immunosuppressant. And so there's been a great deal of interest in immunotherapy. And that's based on PD-1, PD-L1. But this is a new pathway that hasn't really been fully explored as a way, a separate immune pathway that's there for the targeting. And we actually are about to start a clinical trial at Vanderbilt in colorectal cancer, combating the ability of this adenosine to actually bind to its receptor. So that's one example of where the discovery work can increasingly move rapidly towards the clinic.
And then, so the next step you said is a clinical trial. And I guess this is, Jordan, where you come in and try to advance that trial. How does that work? If, for example, Ethan's new Wnt inhibitor he wants to put into patients, or a biomarker that's predictive of what might happen for a patient, if they want to translate into people and they think it's going to be valuable, what happens next?
So it's actually one of the key areas of research is that transition from the laboratory to the clinic. And that's actually the experimental drug program I run, the phase 1 program. We divide the clinical trials up into phase 1, 2, and 3, but we've really blurred the lines across all those phases to try and learn more in earlier phases. But the basic thing is, first we have to prove that the substance is safe. We do that mostly in organ models, so we may test it against liver cells growing in a plate. But sooner or later, and please don't hate us for that, there are some animal tests that have to be done to see that it's safe in animals to get an approximate dose based on their height and weight. Obviously, mice might weigh a little bit different than most humans. And if you have mice that weigh about the same as a human, I think you should get scared. We then take that dose and we choose a dose well below what was dangerous to mice in particular. And we start with that dose and in humans, and then we try to escalate the dose, bringing it up in a small group of people at a time for safety, because we don't want to give the drug to 50 patients and then find out that it was really toxic, but that side effect happens a month later, and you know, all those people are about to face a really horrible side effect. So we only treat a few people at a time, and we watch them real carefully with a lot of laboratory tests, a lot of extra what we call pharmacokinetics, because we try to understand how the body breaks down the drug, what happens to the drug. There can be extra special scans. There's all kinds of tests done to try and learn as much as possible about an agent. When a doctor gives you a pill and tells you that the pill, you have to take it every day, that's based on what we call pharmacokinetics, understanding how the drug is broken down, how long it stays in your body, and all that is done in the phase 1. So we test that, and then if the drug is safe and can be administered at doses that look like they would be effective as based on the laboratory data, then we bring it to phase 2, where we test it in a specific disease, for example, colorectal cancer, and we see if it works against that disease. Certain drugs may be better in combination than single agents, so we may not do a phase 2 till we prove combinations are safe as well. So that's how these drugs are brought into humans, and the people who go on the trials are obviously hoping that they'll work. We are too. But they're also knowing that they're taking risks with new drugs in hopes that they're going to help change the future for cancer patients beyond them.
So I will interject at this point to let everybody know that if you have questions, you can, for the panelists, please go ahead and put them in the question and answer section, and we'll try to get to those questions. So to return to this, once the knowledge from the basic scientists is being translated into these clinical trials, is that the end for the basic scientists, or do they still have a role to play at that point as well? Ethan, perhaps you might talk about this. For example, if your Wnt inhibitor is being used in clinical trials, are you out of it at that point, or you still have a role to play?
Yeah, I mean, we, of course, you want to identify those individuals that are more that could be most benefit from the drug. And it turns out that you have this model of how it would work, but sometimes when you actually treat patients with these drugs, everything goes out the window, and maybe certain patients respond and other patients don't respond as well as you would expect. And so you have to go back to the laboratory and try to understand how the drug works, or maybe your model is incorrect. And just to give you an example, there has been a lot of excitement about certain class of RAAS inhibitors, and Bob can maybe tell us a little bit more about what he's doing with RAAS inhibitors. But there are these drugs that will target certain mutations in RAAS, which is a well-known oncogene. And it turns out that not everyone who you expect to respond. And so you have to figure out why they don't respond to the drug. And so so that you have to go back to the lab and try to understand, maybe you're missing something, some maybe basic mechanism that you don't understand. And so and it turns out that a lot of cancer drugs initially, even though they're effective, may not be what you be targeting what you think they're targeting. It's well known that there are drugs that are very effective, but somehow they don't. The actual target that you think they're working on isn't what happens in the body, and you want to know that because you want to make even more effective drugs to maybe more selective that can minimize side effects and other things. So yes, there's a lot more work even after the drug is placed in the individual and used for treatment.
I mean, one other example along that lines is Ethan may develop the perfect drug for beta-catenin, but unless it hits the target, then it's not going to work. And one of the strengths of Vanderbilt is to have all of these different strong shared resources that allow us to be able to make that determination. Can we, in fact, find that the target is present in this particular tissue and that the drug is actually getting to that tissue? So there are many ways that cancers can be elusive. We are sharing information, and that's one of the values, I think, of an enterprise like the SPORE, where basic scientists like Ethan and clinicians like Jordan are coming together, sitting at the table and sharing ideas and sharing approaches. And as an example, for the first iteration of the SPORE, we went after beta-catenin, after Wnt signaling. And Ethan came up with a drug that now is being tried clinically in a certain setting. And then we realized that another supposedly undruggable target was mutant KRAS. So Steve Fesick at Vanderbilt has now developed a program, and that was the second iteration of the SPORE, to go after mutant KRAS. And now he's licensed molecules that appear to have some activity to a pharmaceutical company. Because for these SPORE projects, we get about $200,000 a project, whereas it costs millions and millions and millions of dollars to develop a drug. So we're trying to get a foothold, and that's what we do with the SPORE project. And so we took it as far as we could, and then it's been licensed out. And in this iteration of the SPORE, Bill Tansey is developing, along with Steve Fesick and Dan Beecham, developing drugs against MYC, another supposedly undruggable target in colon cancer, but has wide, if such an inhibitor would have wide applications to many other cancers. So it's another example where we're trying to get a foothold, and then if it works, we'll advance it.
I think the proof in the pudding as to the importance of basic research in making drugs in cancer is the fact that the founders of the most of the biotech companies in the US have come from university performing basic science. I'll give you examples: Genentech, Vertex, Regeneron, Moderna, which I'm sure many of you now have heard of. And so I think it reflects the fact that medicine in the 21st century is basically cutting edge research, and it's fundamental research. So understanding fundamental biological processes is what can get you to the next treatment. And that's why basic research is so important.
Okay. So I actually have a question that is interesting to me. We heard earlier that in response to treatments, not all the cancer cells are killed. Some remain dormant, some are resistant to the treatment. And this gets at the idea that the cancer cells are diverse. There's heterogeneity in those cancer cells, and that allows them to survive these treatments. And I'm wondering, is it the mutations in these different cells that is important, or is it the microenvironment in which they're in, the specific niche that those stem cells are sitting in that allows them to survive? What is allowing that resistance to be there? Do we know the answer to that, or is that a question that we still have to research? Anybody want to take that one?
I think I'll make a very quick answer and then maybe pass it along to somebody else. I think this is a question for all cancers, not just colorectal tumors. But I think the answer is kind of all of the above. So some tumors have a mixture of cells in which different pockets of the tumor have a different combination of mutations. But then there's also things like non-mutation driven heterogeneity. So there are layers in the genome, non-genetic or epigenetic changes that can also affect gene expression and response to drugs. And then the microenvironment can also influence both accessibility of drugs and things like the growth factor milieu of the environment. But I think that maybe someone like Bob or someone else could speak more specifically to, colon cancer heterogeneity in response.
I mean, I could say, make a comment about one of the targeted therapies that has been used in colon cancer with modest activity in terms of being a single agent and better in combination with some of the regimens that Jordan has talked about, and that's cetuximab, and that's an antibody that blocks the EGF receptor. And we've found that when we do genetic mutational analysis, which is increasingly done on many solid tumors, that those tumors that have a mutation in KRAS do not respond to cetuximab. So there's an example of finding a mutation that isn't predictive of something that might work, which we're always looking for, but a mutation that tells us not to use a drug, which can be beneficial because then those patients will avoid the potential side effects of using that drug. But what we've done at Vanderbilt is we've been able to now identify new mechanisms of resistance to cetuximab that are non-genetic. One in particular is related to a long non-coding RNA that we found that by circumstance and diving deep into the mechanism, we determined that it was upregulating Wnt signaling by downregulating 5 Wnt negative regulators. So it then brought us back to the Wnt pathway, which Ethan is an expert in. And what we found is that if we now add a Wnt inhibitor to cetuximab in that particular set of patients that have developed resistance to cetuximab that are independent of KRAS, that now those patients will respond. So, there are many layers of this, but the fact that we have experts in so many different areas, and having everybody at the table can allow us to compare data, share results, and then try to devise additional beneficial therapeutic strategies.
So, well, as we're approaching the end, I want to just hit that last point. So what do you each think is the next frontier here? How is the basic research that's being done today going to actually change our outcomes and our therapies in 5 to 10 years? What do you expect? Do you have high hopes that obviously that what you're doing is going to be useful, and what kinds of expectations should we have? Does anybody want to take that?
Well, it's easy for me to answer that we don't know. Of course, the thing, if you're looking on the internet, that's been big in the area of cancer treatment has been immune therapy, and for whatever reason, 99% of colon cancers are resistant to immune therapies, and so we consider figuring out how to make the immune system work in colon cancer kind of a new holy grail for therapy. And there are a lot of trials going on trying to overcome the resistance. There was a small group of colon cancer patients that are incredibly sensitive to immune therapies, those with what we call microsatellite instability. But the rest, we have yet to figure out the key element that's missing. Somebody asked in the chat about microenvironment versus genetics versus other things that might be driving the cancers, but this is where the basic scientists are the key to us, figuring out what the drivers are, how they interact, and for people who are bioinformaticians, you get multiple genetic pathways, and understanding how these genetic pathways interact, why one person responds to a therapy and the other one doesn't, possibly because they have 2 mutations, the other person had 3, very well could be an important component. And that's going to require a lot of computational skills that I definitely don't have.
Yeah, certainly this is a big topic. There's many researchers at Vanderbilt working on it from many angles. We heard today from 4, and from both the very basic science to the translation. And I really appreciate your time today, everybody, for contributing your knowledge. For the people who are listening, we will have video. We've recorded this video, and we can make this all available to you if you want to share it or listen to it again. We also can send you resources to all of the attendees on this topic. And I'll just close by emphasizing that Vanderbilt is a very unique place doing this type of research. We have very basic scientists, everything from chemists to cell biologists, who we heard today, to physician scientists and clinicians. And this environment allows us to really have a good perspective. And of course, we're working with all of our colleagues throughout the country and throughout the world to improve the outcomes of this disease, and also to prevent it. So I'm hopeful that the trajectory is good for this disease. We've been making progress. Certainly, we need to do better. But I'm very hopeful and thankful for all of the support that the public gives to our research to be able to do this and to make improvements. And so thank you to all of the panelists. I appreciate your time, and I think we will end it here.